All right. Thank you, Chip. You see my screen now?
We're seeing your screen and hearing you.
Okay. Well, thank you for the opportunity to talk to the council today and everyone else who's interested. So I'm going to present a little bit about what you just mentioned, the fish acoustic detection algorithm research, which is a deep learning app for Caribbean grouper calls detection and call types classification. So we're going to go over different aspects of acoustics and fisheries and how FADAR can be applied. So before we start, I just want to acknowledge the FADAR team, in particular Michelle Charrer in Puerto Rico, who's been really the person who set the stage for this research, who got me interested in the science of fish acoustics, and who provided a lot of the data that's been used to create FADAR. I also want to acknowledge Ali Ibrahim, Dr. Ali Ibrahim, who started as a PhD student on this project, then graduated and continued as a postdoctoral research associate, and then is now a research engineer at FAU HBOI, and is also a staff in the Department of Electrical Engineering and Computer Science at FAU. And then I also want to acknowledge Caroline Woodward, who was one of my students, who is now a research associate, and has been helping a lot with the validation, testing, and application of FADAR. So this is the outline of my talk today. We're going to talk a little bit about passive acoustic and fishery science and how passive acoustics is relevant to fishery science. How we go about associating sound to fish species, which is one of the biggest challenges. How we use fish sound to discover spawning aggregations and how it's been used. How do we measure sound and how do we listen in on those fish and those aggregations. And then how we go about doing the sound analysis, and in particular that will take us to the main topic of the presentation today, which is FADAR, and how FADAR results and what we get out of it can be applied to fisheries research, and then we provide some conclusions. So what about passive acoustic and fisheries science? When you look at recreational and commercial fisheries species, many of them are highly soniferous species, including the cod, the drum, and also the groupers. And so how do fish use sound? They use sound to communicate with one another while feeding, mating, or being aggressive, defending territories. But they also make incidental sound while feeding, swimming, and with other behaviors. And unlike, the idea of the silent world of Jacques Cousteau, our estuaries and seas and oceans are teeming with sounds of marine life. And so one of the challenges of studying fish in the ocean is to locate them, and sound, passive acoustics in particular, provide a way to do that. And so the advantage of that method is that it's a passive approach. Basically, you're just listening for the ocean sounds, and instead of using artificial sounds to track animal or find animals. So what are the benefits of passive acoustic? It's a non-optically way to observe fish activity and distribution. It is a non-invasive and non-destructive observational tool. It offers the capability of long-term monitoring as well as remote monitoring. And it can also be used to not only monitor or survey or look for the presence of certain species, but also assess the presence of other sound sources, in particular those associated with human activities. And so coupling passive acoustics with conventional fishery sampling techniques can provide a powerful approach to fisheries research, and we're going to see how this is used and the different steps we have to go through to use passive acoustics. The first is to associate sound to fish species. So we know about 800 species of fish that are soniferous. About 150 of them are found in the Northwest Atlantic. And Among them, some of them are the most abundant, important commercial species, including the codfish, the drum, the grunt, fishes, the grunts, grouper jacks, catfish, and there are also invertebrates that produce sounds such as mussels, sea urchins, white shrimp, spiny lobster, American lobster, and perhaps squids. Passive acoustic have been used for 60 years in fish biology. It's been used in fishery surveys, and today it's routinely used to determine habitat use, delineate and monitor spawning areas and aggregations, and the study of fish behavior. The way this is done is by basically submerging a hydrophone in the water, listening to the sounds, and using signal processing to identify the sound to specific species. But the challenge there is, how do you match a sound to its source, so basically to a specific species. Sometimes the sound they produce can be really overwhelming in the environment to the point that it might interfere with human activities as well. So what we're gonna do now is basically show that before we can label the sound as belonging to some species, we have to do what is called sound truthing. And there are different ways to do that. Basically, two ways. One is based on captive experiment, where the fish is kept or held in a tank or an aquarium, and the other way is in the field. They both come with advantages and their caveats. If you're in the tank, you may have a lot of reverberation of sound. You may have the fish behavior may change, and sometimes you may hear sounds that might be slightly different from what you would hear in the wild. When you're in the field, on the other hand, the challenge is to match the sound to the species or to the animal that's producing the sound. And so the other thing is to identify, the other challenge is to identify the proper location to put your hydrophone, because you may be, you may think that you're close to the habitat of where the fish hang out, but as you know, fish move around. If you are at a spawning location as well, they may be coming and going, and if you're not close enough to the source, you may not capture the sound properly. So despite all those challenges, we've been able to link aggressive and spawning behaviors of some fishery species to the sound production using a combination of in situ and tank studies. For instance, the case of haddock and red hind grouper, some of the courtship sound and mating sound have been recording in the tanks. And when it comes to in situ observations, in particular in tropical waters, environmental conditions allow for the use of both acoustic and video technology, so you can really tag a species to a sound. Once this association of sound to species has been made and behaviors have been identified, passive acoustics provide a rapid way of establishing the spawning component of essential fish habitat. Okay, so now we've been able to associate, sound to a species. How do we go about discovering, using sound to discover spawning aggregation? First, we need to understand the function of sound production. In most of the species, the fishery species, the most predictable and robust sounds are associated with production, which is a good thing for us in terms of being able to find them and study them. And in most of the species, it's mostly the male that produces sound, and it's been acknowledged that sound production during courtship and spawning is a way to synchronize. Spawning is a way to entice female and male to release eggs at the same time, and which facilitate the fertilization of the eggs. But females have also been heard to produce sound in some other species, such as the red hind. So some species have, if you want, a repertoire that may be more elaborate than others. And we're going to see that those repertoires may be also associated with particular behaviors during the aggregation process or different stages of the courtship. So in regard to that, as we were saying, you know, most sound production is associated with aggregation, in particular with the groupers. Most of the courtship associated sound or the sound that we hear at aggregations are only heard at fish pooling aggregations. As we go about, you know, studying those aggregations more and doing captive experiment, we actually are able to discover a more diverse repertoire of sounds that those fish are able to produce. And for instance, it's been the case for the red hind and also the Nassau grouper, which both exhibit agonistic sounds and sound associated with territorial defense. So one aspect that is important to consider when we think about sound produced by fish is the sound production mechanism, because it may dictate the type of sound that the fish is able to produce and what to be expected, and also the diversity of sound that one species can produce. So I'm going to go over different species to show you a little bit the differences in the sound producing mechanisms. If we look at the cyanids, the sound is produced by a sonic muscle, indirectly or directly vibrating the membrane of the gas bladder. So for many species, it relies on the expression of a sonic muscle that is connected to the gas bladder, and which ended up producing a sound, the vibration, a sound produced by the gas bladder that vibrates at the same fundamental frequency as the muscle. When it comes to gatid, the cods, what people have observed is that there's actually a sexual dimorphism in the sonic muscle in the adult, where the male have a much larger muscle than the females. And in addition, the muscle undergoes seasonal maturation in concert with gonade maturation. When it comes to groupers, the only grouper species for which sound production mechanism has been studied is the Nassau grouper, and it involves a contraction of a sonic muscle that is connected to the vertebrae and swim bladder. So what we see here is there are different, if you want, settings in terms of how this muscle is set up in the fish, and that will basically determine the kind of sound and the diversity of sound that the fishes are able to produce. So we're going to go over basically what those sounds look like for the different species that we talked about. And what I'll show you here is basically a series of oscillograms associating with the sound produced by sea trout, labeled A, silver perch labeled C, and the black drum. And you know, back in the days, people were focusing more on identifying the calls using those oscillograms, and as you can see, they are pretty distinct when it comes to from one species to another, right? If you look at the sea trout, you can see that the structure of or the pattern of the oscillogram is different from the black drum, and also different from the perch. But with time, you know, we started looking more into the image of the sound, which is produced by a spectrogram, which shows the variation of frequency over time, which is shown here for the silver perch on the right side, and for the black drum in color below. What you see here for the black drum is actually a black drum chorus, which means like several fish are calling at the same time. And then, so what happened is that information was used to really characterize sound specific to each of those species. So you have on the left rigfish recording in Pamlico Sound in North Carolina. So you have at the top of each pairs of images, the top is the oscillogram, the bottom is the spectrogram. You can see basically, so it's the redfish on the left and the citron on the right. In the top is in the captive setting, and the bottom is in the natural setting. And you can see that there could be slight differences between sound recorded into captivity and recorded in the wild. Here's another image showing an illustration of the spectrogram for the silver perch, and also the distribution of the different concentration of fish based on the distribution of sound in Pamlico Sound, using sonobuoys that were drifting or attached to the bottom. And that's basically using a drifting hydrophone in the water, or driving around with the hydrophone allows you, based on the sound signal that you capture or you hear, to identify the location of the fish. And again, you can notice here the slight difference in terms of based on the captive setting or the natural setting in the sound, in the oscillogram and in the spectrogram as well. I'm showing you here now some spectrogram and oscillogram from the grouper species. So we have the top here, RH1 and RH2 that are related to the red hind grouper, then YF1, YF2 related to the yellowfin grouper. You see two sound types here. You can see the oscillogram associated with those. And at the bottom here, you have the black grouper. Some of those sounds consist of different components that we call pulses or tones, or tonal sound, that they're expressed by this sort of sweeping change in frequency, while pulses are associated with the vertical bars that you see here. And you can match somehow the oscillograms with the image in the spectrogram. On the right side is the sound produced by the male haddock, and at the bottom here you see the spectrogram produced, the sound produced by the Atlantic cod in the southern Gulf of St. Lawrence, the top against the oscillogram, at the bottom the spectrogram. So depending on how you process the signal, you can get a very clear image of the sound, which also, of course, depends on the ambient noise, because this sound can be sort of not smothered, but can be nested into the noise and less discernible in the spectrogram, which also will make the oscillogram a more noisy signal. And then here, we have all the groupers that are basically the main species that we have focused our attention to. And I'm going to play the sound, and let me know if you hear it or not, but so you can get an idea of the differences. I'm going to click on the images, but for some reason they don't play in the order of the image, the way I clicked on the images. So I'm going to let you know what it is, if you can hear it. Can you hear that?
Yes, we can hear it.
Okay, so that was the red hind grouper. That was the yellowfin grouper, which is my favorite. This is the Nassau grouper. And that was the black grouper. For the black grouper, it's a little bit more challenging because it's a simple boom. I think anyone, just doing boom, it's enough to make you realize what kind of sound it's produced. And then what I wanted to give you a little bit a feeling of is when you listen to the ocean, the kind of noise that you may hear and how all those different sounds may overlap. So what we created here, it was a little project that we had with Bob Currier at GCUS and Stuart Fulton. It was to create a sort of fish radio, and what you see here is the spectrogram of all the sound that are actually playing, where you hear you hear a number of toadfish actually, and a Nassau grouper towards the end. So we could play that for a while if you wanted. But anyways, that gives you an idea of the different sound, and then this fish radio gives you an idea how they may overlap, and then the challenges in that we have as someone who's trying to figure out, all the different species that are producing sound. Okay, so now we're going to see how we go about identification of spawning aggregations. So some species, in particular cyanids, have been known to produce sounds for centuries, and the association of the sound production of cyanids with spawning has been known for nearly as long. People would just walk to the dock or from their house on the water, could hear the sound of the fish on the water. And so the way we go about, looking for those aggregations is basically the simple method is by, scanning the water, deploying a hydrophone in the water on a boat or now on a mobile platform, and based on information given by other scientists or local fishermen, look for the presence of fish, location that might be known, or basically drift around while listening for sound. And so this kind of technique basically, which is, relatively simple, allow for spatial and temporal isolation and identification of species, specific sound produced by cyanids, in this case, for all the species listed here, including groupers as well. And so when you do that, and you're able to identify those locations, then you can set up, as we mentioned earlier, long-term monitoring that will provide you with the specific pattern of fish sound production in order to understand the drivers of those patterns. So for instance, here in the case of cyanids, you can see that on the graph here, you have the number of calls basically produced by red drum, spotted sea trout, wigfish, silver perch at different time of the day. And what you have, the vertical axis is the drumming index, so basically how many sound are produced. And you can see, for instance, in this guy, for this species, that there is a pattern that where a peak sound production is associated with the evening hours for at least 3 of the species here, but also the morning hours for the silver perch. And then if you do that over a longer time period, you are able to identify the spawning season, the movement pattern of the fish, whether they leave the aggregation or they come back, and many other aspects of the spawning aggregation dynamics. And so when you start looking closer, even at other species, you start realizing that, for instance, for the cods, they have 2 periods of time production, and one is associated with spawning. And during that period, you find that most of the calls are produced during the evening hours and in the night, while when they, in the other time period, when you have calling activity, which corresponds to the fall, what they call the aggression period, calling happens mostly during the day. So those patterns can tell you a little bit about the fish behavior and when you should focus your attention when it comes to spawning, or when you should look for them. So when it comes to groupers, again, most of the sound produced by groupers and the diversity of sound here heard at spawning aggregations are only produced during spawning season. So which means outside the spawning season, they don't produce the sound. So that gives you a very specific window, but also a very sort of facilitate the way you'll be able to target your survey, because you know that when you hear those sounds, it means that the fish are preparing to aggregate or are aggregating or are, in spawning. And so using the sounds, you can actually create a time series of sound production. And by studying these time series, you can actually infer some relationship to environmental drivers. In the case of the Caribbean groupers, and for instance here, red hind, at Red Hide Bank, South St. Thomas in the Caribbean, you can see that peak calling and spawning, which has been spawning, and so basically spawning irrigation has been queued to is basically queued to the full moon cycle. And what Rick Nemeth and his team realized, they basically realized that peak aggregation, spawning aggregation, was related to the first full moon after the time between the first full moon after the winter solstice. And he was able to predict that which month would have the greatest aggregations and therefore the greatest spawning effort. And you can recover the same trend by looking at the sound production time series. Sound production has been used to locate and search for unknown spawning habitats. That happens, for instance, for cod in Norway, where they think that it was known that the fish used to spawn offshore, but using sound, they realized that there was an inshore spawning aggregation. And when it comes to groupers in the Caribbean, what listening to them has told us is that many of those sites where, for instance, people have been mostly targeting red hine, which is one of the biggest commercial fisheries in the Caribbean, it showed that other species also aggregate to spawn at the same location, whether at the same time, whether at other times, but at the same location. And I'm showing you here, I'm not sure if the council ever seen those video, but it is an experiment that we conducted in 2011 at Granitic Bank in St. Thomas, where we basically surveyed with the ROV the spawning aggregation there of yellowfin grouper, where we saw actually the presence of Nassau grouper as well. It's a huge aggregation with at least between 8,000 and 10,000 fish that we surveyed. So, while monitoring for one species, searching for one species, you actually find other species, which allows you to understand the importance of those spawning habitats for multiple species at the same time. And then, I call it here the get lucky factor. In October 2015, we were testing the passive acoustic monitoring system that we installed on the wave glider. And we wanted to test it near the staging locations of coral grouper of Jupiter in Florida, where you have those famous wrecks that are known for large aggregation of coral grouper before the spawn. But instead, we made a mistake on the waypoint that we put in the glider. We sent the glider, as you see, about between 2 and 5 kilometers off the staging location of those groupers. And what we found there is that the groupers were actually out there producing sound in a way that is very similar to what we saw are the spawning locations that are monitored acoustically. So in some ways, we've discovered, that the spawning location of coral grouper in that area may not be at those wrecks, but further away from that location. Okay, so how do we measure sound? I think it's the most important part. How do we get good information, good data that we can make good use of it? And for that, we need to consider different factors. We need to consider the sound level. So how loud are the fish? We need to, therefore, that will dictate the type of hydrophone and the hydrophone sensitivity that we make use. So basically, if a fish has a source level of about 150 dB, we want a sensitivity that is low enough in order to encompass the entire extent of the entire amplitude of the signal that is produced. The other important aspect is the sampling frequency. So if your fish is producing sound as, let's say, at 4 kilohertz, then you need to at least have a sampling frequency of 8 kilohertz in order to resolve the 4 kilohertz frequency. And so you need to account for, the type of sound, the frequency range in the acoustic in the soundscape that you're trying to capture, and which means that it will determine the amount of space that will go into your memory for each file that is recorded. So the higher the sampling frequency, the more data basically is collected by the hydrophone. So the more space limited you will have, your memory is going to be. There are different ways to also use hydrophone. You can use a single hydrophone if you're able to locate the aggregation, the spawning aggregation, properly. So making sure that you're going to be at the place where the fish aggregate to spawn, or you're going to be at the location where the fish transit to go there, or maybe you're going to be at the place where the fish stage. So what that tells you is that if you have a hydrophone array, the advantage of that is that, and if there are time synchronized and and sometimes you have to go back in the field and do what we call the hammer bank to basically use that as a reference to synchronize the time of your hydrophone, because if they're not on the same time, so if it's not an array that is controlled by the same recorder, then they may drift. They may drift in time differently from each other. So it is a challenge to realign the time of the hydrophones. But if you can do that, then you can localize the sound. And if you localize the sound, then you can assess the movement of the fish within the array and around the array. The other thing that you can do is you have to determine is therefore the sampling the cycle of operation of your hydrophone. If you use a continuous recording, usually you can only do that with the current capacities for only a week long. And otherwise, if you want a cover period of several months, then you have to use what we call duty cycles, where, for instance, as we used in the Caribbean, we record for 20 seconds every 5 minutes, and you can extend your recording up to 6 months. So what comes out of that is usually a large amount of data of the order of 1 gigabyte per day, depending on the duty cycle. And in some instances, some companies have a couple of video recorders with hydrophone, which allows you to basically sort of validate the sound that is produced and associate the sound to a species. Yeah, and the other thing I wanted to say is in terms of properly localizing the aggregation, you can also set up an array hydrophone around the habitat where the fish are observed to be. For instance, something we did in the Cayman Island, where we set up an array along the entire edge of the shelf, where the fish are shown to move back and forth. And in doing that, you're able to actually identify the location where they aggregate to spawn, the location where they transit, the location where they stage. Because if you account for the sound propagation, how far the sound propagates in the water, you may see that when the fish are really move to one place, one location, and that's the spawning side, you may not hear them further away. So you know that they've all moved there. Anyways, so you can do a lot of things with sound using static hydrophones, but we can also use autonomous platforms with passive acoustic monitoring system, which is the case of the wave glider that I talked earlier about, which we developed here at Harbor Branch, and has also an embedded detection algorithm that basically allows you, that tells you in real time when courtship associated sounds are detected, which allows you to maybe mobilize your team to verify whether it's an aggregation or a staging place. We did a little bit of that in the Caribbean, as as an evaluation of the approach back then. All right, so when you collected all that data, now comes the time to analyze that data. So the way we've been going about this is to basically sit in the lab and listen for hours and hours to the recording with someone specialized in the different sound as I played them. So basically, these are the one that you try to recognize, and to do that, there are a bunch of software or tools available that allows you to basically pull out what I showed you earlier, which is the waveform or the oscillogram of the sound, if that's your way to go about it, but also to create spectrograms that allow you to basically visualize the sound. So we have Audacity, Adobe Audition, Raven from the Cornell Lab, and also Ishmael. And then based on the way you identify the sound, or whether you want to go through looking at features, acoustic features in the spectrogram, or acoustic features in the oscillogram or the waveform, you can basically look for the features that best represent a particular sound, and then use that to create a notably algorithm that will specifically identify those features. So we calculate them for each sound sample or time period that you select, and provide you with a result. As you can imagine, it is a tedious effort, and there's not really any method that's automated for a variety of species. It's been done for specific species. It works pretty well, but it's a pretty tedious process. And so given the challenges that are there, we came up with the idea of developing an automated method based on machine learning that we call fish acoustic detection algorithm research, FADAR, that was specifically built for the detection of Caribbean groupers, because we were working at the time, and we're still with the Caribbean fishery biologists that were studying spring irrigations over there. And so now we're going to get into, FADAR, and I'm going to provide you the information and description of the FADAR algorithm. So FADAR is a CNN-based deep learning algorithm. It was designed to identify and classify the sound of four Caribbean grouper species that that I presented earlier. So namely the red hind grouper, the black grouper, the yellowfin grouper, and the Nassau grouper. And in addition to that, we also detect one very common species that we found at those aggregation, which is the squirrelfish. And also we made it so that we can also identify the sound of surface threshold, which allow managers basically to infer fishing pressure and poaching, etc. The other thing about FADAR is that we don't work directly from the raw sound sources. We actually convert the sound to an image. As you saw, that image can really tell the difference between the different calls. And so CNNs were actually developed as image recognition algorithm, and so that's where the application came from. This is the architecture, the overall architecture of the model. So its general structure consists of a main CNN model for all sound classes that you see here: the red hind, the Nassau, the black grouper, the squirrelfish. And then we have, and that is done by the main CNN model. And then once you've done that, we have specific call types, some models that will identify the call type for the red hind, the Nassau, and the yellowfin groupers. So how do we get about creating FADAR? Well, when it comes to machine learning, it comes to training, labeling data. Okay, so taking recordings, cutting them into little, if you want, samples. And here we chose like 2 seconds audio, spectrograms, and we label all of these, and we train the model with that, and then we tested the model, and then on the same sort of like data stream, and then we use independent model. I'm going to show you a little bit the approach to do that. So basically what I want to show you here is where this data came from. It came from the Western Shelf of Puerto Rico, a non-spawning irrigation site. So the thing I want to say is that FADAR was specially trained on data that came from a fish spawning irrigation site in the Caribbean. So basically the data was curated for, and the model is curated for this particular regions and application and data. So we use data from the Western Shelf of Puerto Rico, Abrila Sierra, Bajo de Seco, Mona Island, in the US Virgin Islands, from the Red Hind Bank, Gramanik Bank, Lang Bank, from the Cayman Island, now the Florida Keys, and the waveguided data we use in the Virgin Islands. So there's a little bit of diversity of data here with different regions in the Caribbean, including different monitoring platform as well, fixed and mobile platform. Of course, when you develop this type of model or any model, you have to evaluate its performance. And when it comes to machine learning, it's, testing is done by evaluating quantities such as the true positive, the true negative. So how many times the model is correct, how many times the model is correct being right, is correct being wrong, how many times the model fail at finding true positive or finding true negatives. They are used to calculate metrics such as, so these are really a jargon which is specific to this type of analysis, sensitivity and recall, which is the ability of the model to correctly identify a call, specificity, which is the ability to correctly identify a non-call, so basically when there's noise and there's no call, and then the accuracy, which is the expression that you see here. So I'm going to show the results of the FADAR in terms of those metrics. The evaluation approach that we took is that we use the data set, the specific data set, among all the data that I listed, for model development and evaluation. So if using, let's say I take, the recording Abriola Sierra for the year, let's say 2012, 2013, that would be my training data set. And so 80% of that data will be used for training and 20% for validation. And that's basically the first two. This basically the sequence that trains the model. And then from the same data set, still Abriola Sierra, we use about 40% of the data set for testing, but the data used for testing wasn't used into validation or training. And then once we've done that, we evaluate the model on what we call an independent data set that is constituted of data from different years, different instruments, different locations, and that were never used into the training or validation or previous testing. And so these are the results that we got from the development data set. What I show here is for each class, so red high, Nassau, yellowfin, black, both sounds, goldfish. We have the sensitivity, the specificity, and the accuracy here. So you can see FADAR performance is very good. Basically, those metrics are range between 0 and 1, and so 1 being perfect, right? And then we also tested the submodels for the call types for the red hind, for the Nassau, and for the yellowfin grouper. For the Nassau, actually Nassau, as you may have seen, as you may know, produce more than one call type. There's about 4 of them, but you can really classify them into one which is more tonal than the others that are more pulse associated. So what we did is that the N1 here is the tonal call, and the N2 encompasses the other 3. And this is the result that we got. And then we took FADAR and tested it on an independent data set that used data from the Cayman Islands, from Gramanic Bank, from Lang Bank, and from the Florida Keys as well. And these are the results that we obtained. So you can see that, for data that coming from the different regions of the Caribbean, from different instruments, from different settings, from different locations, of course, from different depths, different sampling frequency, etc. We have a pretty consistent performance across all those data sets. So FADAR is freely available. The app, not actually the actual MATLAB code. So what we released basically is a MATLAB executable that works on a 60-bit computer that requires a little bit of RAM. In terms of data requirement, your data needs to have a sampling rate that is greater than 10 kilohertz. And then in order to operate the app, you need a MATLAB runtime, which is a free sort of operating MATLAB system that you can download. And so if you go to GitHub, you can download FADAR executable directly. Sometimes you may have some issues, and so what we recommend really is to get the app installer, which will install runtime and creates FADAR executable once you download it. So how does it work? So basically, once you've done that, the installation steps, you just click on the app. It will open up a window that look like the one on the left here, and you have basically three options, right? And the first option is the output type that you desire. You have several possibilities. You can process the data and display the data in the app. And I'm going to show you what it looks like. You can export not the data, but the results of the classification into an Excel file, or you can create an annotated video, which allows you to basically verify the detections and the classification that FADAR has made, because it's a label. Yeah, it's an annotated video, so it writes the name of the classification, and you can right above the image of the file that has been analyzed. And also, we created this tool, which is output verification. If you select output verification, what's going to happen is that it's going to create folders for each of the classes. In those folders, it's going to fill them up with little stamps, like images of the spectrograms of all the sound that all the sound that it has analyzed. So basically, if you have 1,000 files of 20 seconds, and every file is split into 2 seconds, timestamp, so you get 2 seconds. You call this images, tiny icons, if you want, that we fill up the folder that allows you to go back and look at basically FADAR performance directly. So you can end up with, I know, 20,000 files, but you can quickly take a look, and I'm going to show you an example of all different cases. So let's start with the in-app results, or what you get basically is on the left, you have the list of all the files that were analyzed, and here you have the different classes. So you have red hind, Nassau, yellowfin, black grouper, skullfish, and for those, I think this is an old image because now you also have vessel sound. And then the numbers here represent the number of calls that were detected. And here it's the time of the detection was made in the files where the calls were, and the files that is here basically. So your files is 20 seconds. The 4 here shows that the call was detected at 4 seconds, and the next call was detected at 2 seconds for the species here. And then this is the time for all the other species too. If you choose the export file, then you end up with a CDS file that has exactly the same format as here for all your classification. And so the advantage of that is that you can actually take that Excel file or CSV file and then plot the result directly over time or over file and see the evolution of your course, which I'm going to show you in a little bit. Here, the spectrogram verification video. So you see at the top, the red hind with given probability. So it gives you, so you can see that what is labeled here corresponds to what you see here, which we have a bunch of red hind calls here, as you can observe. And then this is the output verification that creates those different spectrograms that will be filled with those little images that you can quickly scan through to validate the detection of FADAR. And then, so once you've done that, you chose the type of output that you want, then you have to select the classification model that you want to use. So if you want a general classification that will give you basically that will classify all the classes, and you select classify grouper, and then, but if you want basically look into the code types specifically, then you can select between red hind type Nassau type, and yellowfin type. Okay. The third selection here is you have the choice between analyzing a single file or analyzing an entire folder. So when you, I know in our case, when we work with the data that we have folders with 10,000 files, basically, right? And for a 6 month period, you may end up with 6 folders of 10,000 files. So you have 60,000 files to analyze. And so you can load one folder at a time of 10,000 files or more, and then you click the button start, and there it goes. Okay, so now what I'm going to show you is what we've been able to do with FADAR outputs and how we use that data to understand some of the dynamics of fish spawning aggregation. In particular, we worked on data from Abriola Sierra in Puerto Rico, and we have now a time series that span a period of more than 10 years. So we've been able to look at the evolution over time of the number of calls associated with the spawning aggregation at Abriola Sierra. And what I show you here is basically the translation of FADAR output in terms of the time series of the number of calls over time. So this is in 2011. And that basically what we're trying to do here is to establish a relationship between fish sound production and biomass. And this is what you see below is so the same time series that was created for 2019. And you see a difference, huge difference between the two is that the number of calls between 2011 and 2019 has doubled, right? We reached about 30,000 calls in 2019 during the main aggregating month, and it was less than 1,500 in 2011. And if you look at previous studies that were done there, particularly by Roll, by Team Roll, it showed that the number of casts increases with the number of individuals, although that relationship may not be consistent all the time. Every time they went in the water, you know, these fish may move around, and you may count less from one day to another, but overall the population may be the same. So, this is an interesting fact, because what this might tell you is that there was a significant increase in the population. Right. So what can we say about, so I'm going to go further into, now you see two curves, right? You see one dash curve, which is associated with one call type, and the solid line, which is actually associated with another call type. So now we're going to go into looking at how the relationship between those call types has also changed over time. And for that, we're going to base our analysis on the work of Zayas and Tal in captivity. And what we're trying to do here is to assess the relationship of fish sound production to sex and maturity stage. And so what we know from historical observations is that for red hand groupers, that males arrive first at the aggregation to set up their territory, and it shows basically it is associated with an increase of the H2 calls as spawning season starts, right? That's what we see going on first here, right? The first search peak, and that calls, that's the first one that starts increasing and becomes significant than the other one. Later on, based on observations, females arrive and male-female interaction increase through harem formation, right? And what we see is that we see an increase of LH1 calls, which ultimately surpass the LH2 calls, okay? And this is only seen for the red hind aggregation at ALS for the period 2013-2017. And so it begs the question as to whether female numbers increased over that period, which led to an increase of H2 calls near spawning time, and which means that call numbers increased over that time period. But what happened is that after 2017, RH2 calls were never higher than RH1 calls, although the highest numbers of calls were recorded. So something happens at the aggregation where you have the shift in the ratio between those calls. So I'm showing you here what happens in 2013. You see RH2 call picking up right at the peak of the spawning, in 2013 all the way to 2017, where we have this slight difference here. But after that period, starting in 2017, this no longer happens, all the way to 2022, which I'm not showing you here, but we're actually working on that. One of the things that you can notice is that previously we looked at the evolution of daily call numbers, but you can look actually at a higher sampling rate. And what we did here, you can also quantify daily and seasonal patterns, and also associated those changes to maybe environmental effects such as climate change and warming waters. So here, what I'm showing you is the 4 hourly block time series of call numbers labeled by callers in the period 1 to 16 days after the full moon, which is when we see the significant increase in call numbers, and in particular during the period 2013-2017, the peak and the passing of the number of RH1 calls, which is associated based on other studies with peak spawning time. What we see here in the callers, 16, 19 hour, it's basically when the fish are known to spawn at sunset. We see a peak in calling activity. We see also of the RH1 call here, right? And that RH1 call becomes significant, starts increasing over RH2 call starting around the period 7 days after the full moon, and then declines rapidly, 16 days after the full moon. So that was in January 2015. In February 2015, you recover the same pattern again. It's a little bit later in the season. But when you compare that from one year to another, you can see significant changes, for instance, in that pattern and when peak spawning activity happens. So we don't have an answer for that at the moment, but it's, using that type of information, it's the type of processes that you start realizing. So being able to build time series at any temporal resolution of call numbers can provide information to internal variations in the timing of the aggregation, peak spawning activity, and also duration. Okay, so we know here, and you see in February, again here, it didn't start as late as it started in January. So what does it mean in terms of spawning activity, population structure, etc., but also environmental factors and variation? So based on that, we can make a number of assumptions. This red hind example suggests that the red hind population at ALS grew and that its population structure most likely changed. So for example, if the number of RH1 calls has been lower than the number of RH2 calls since 2018, it might suggest that many of the large females became male and there would be less females to call and more males fighting for territories, which means that the RH1 call would be really much higher most of the time, as we're seeing now. So I'm not saying it is the case. It's an hypothesis, and as you can see by looking at this data, a lot of hypotheses can be formulated which require in situ verification. And for that, we have to rely on other technologies, and you see here the value of using passive acoustic monitoring that complement other, fisheries independent observations. And I'm thinking here, diver surveys, but also autonomous vehicles and ROVs, which could give you an idea of the, if you can count them, and if you can identify male and females usually by looking at the distension of their bellies, etc. And so another aspect of FADAR that I want to talk about here is that it can be used for facilitating monitoring for local communities. And FADAR can be really operated by anyone with a little bit of training. We've done that several times. We've provided FADAR to local communities in Mexico, Kobe, a local NGO. We also provided FADAR to the Department of Natural Resources in the Cayman Islands. And so what FADAR does, it enables them to follow trends in their local FSAs and assess the success of their conservation measures. And I'm going to show you some examples. And it provides, in addition, visuals to fishermen of the dynamics of the FSA that they contribute to protect, which reinforces their engagement. And so I've been collaborating with Stuart Fulton at Kobe, and he has basically, we had a little workshop where we trained some locals to use FADAR, and since then they've been using it to look at their spawning aggregations at different sites. So you have Charlac here, you have Punta Island, and you have San Juan, and you can see in terms of numbers, number of calls that may not be necessarily associated with the number of fish, but it could very well be the case because what they try to do is to verify, have divers do surveys, even the fishermen themselves in water, and sort of correlate the population numbers with the number of calls that they see here. So that was in the season 2021, the top, and 22-23, 21-22 and 22-23 at the bottom. And you can see that there are significant differences between locations which are associated with the different number of fish there. But there are some caveats to all of that that I'm talking about here, which are all the challenges when that comes with using FADAR and using acoustics in general. Although the accuracy is high, FADAR is sensitive to errors in the data quality. So if it's very noisy, but also it can be mitigated with the type of data used for training and how you label them. But also what we notice with recorder defects. So some recorders may have some issues. You end up with a noise floor in the recorder. So basically a buzzing that will basically throw off the analysis because the image of the spectrogram of the call will be completely different. The other thing is that, there's no sort of self-evaluation in FADAR. The misclassification rate is manually estimated, but there's some simple ways to do that we've applied. And then other things is that we've realized that when it comes to the groupers, you observe this variation of call rates and call type rates, and those calls are some years very well discernible, right? And at some periods, you end up with what we call choruses, where you have overlapping of so many calls that they become undiscernible in the spectrogram. Therefore, you end up with a fuzzy image that the classifier cannot classify. And so sometimes, because of choruses, our interpretation of call type presence absence will be biased, because you may have a peak in calls that turns into a chorus, but actually you can actually detect them individually in the spectrogram. So it requires several things. First, understanding that this might happen, understanding the call behavior of the fish, how it might be associated with the number of calls, number of fish, and also what we realize, how it might be associated with different populations. We have regional specificities. For instance, in Mexico, unlike the Cayman Islands, Puerto Rico, and the USVI, Nassau grouper exhibit a coercing behavior, something we've never heard in other islands. So we discovered that by noticing, talking to Stuart at Kobe, saying, yeah, we saw a lot of fish in the water, but we only have a few detections. And so we started looking into it, and we saw courses, basically courses that look just like the one produced by cyanids, such as the red shrimp. Quite incredible. And FADAR is prone to errors, and what it means is like you need to understand your local fish behavior to better assess what the type of results that you can get from FADAR. FADAR future, as I was saying before, trusting FADAR, one needs to understand the data at hand and the specificity of the region, in particular the fish calling behavior. We could add other classes to FADAR, other species produce sound, but it's a very intensive process. It's time consuming, it's data hungry. You have to label the data yourself. You need a lot of data. And then sometimes if you try to get into call types, you may have to basically look for the best, deep learning algorithm for your particular application, those particular calls. You could go with, general classifiers that through a method called transfer learning that we've used, but I didn't say, but FADAR was basically the product of the development of various methods over time that we publish in a few papers that allowed us to really narrow down, what was the best approach for all those different classes and what were the best submodels for the different call types, etc. So there are other ways to go about that, making this approach more diverse in terms of the call types, the call type diversity that we could use. There are, many new papers that came out that specifically address this type of issue using unsupervised learning and clustering, where you don't really know the sound sources, but you can use a clustering approach that will identify on its own in different frequency bands all the different sound sources, and then once you have them, you can label them themselves. You can basically do what we call a supervised clustering or supervised learning. So you're able to go in, make some changes, improve the clusters if you want, and then use that to train a model to do the classification. It's a pretty flexible method in the sense that you can add or remove some of the clusters depending on what you're interested in. And so that brings us to our conclusions today. Now we have low-cost, high-capacity digital recorders that are available. We have autonomous underwater and surface platforms with real-time PAM capabilities that are available that will allow you to basically search for those soniferous aggregations. But the sheer amount of data collected can be intimidating. That's why we develop a classifier like FADAR, which now offers the possibility of analyzing large acoustic data sets in an efficient and timely manner. One can assess the presence of multiple grouper species. So that was the main objective of FADAR, right? When we started realizing that those spawning aggregation sites were not only used by one species, but by multiple species, including all those 4 grouper species, that led us to basically focus our attention and include other species all at once in the classifier. I've seen that call rate dynamics is linked to fish behavior, sex ratio, and population structure changes that need to be assessed by other independent methods. And ultimately, if a model of population dynamics at the FSA can be built solely based on the acoustics, then the evolution and changes occurring at the FSA and maybe at the population scale can be tracked, if not predicted, over time. So I think this opens the door to doing things that we wouldn't be able to do before, in particular predicting the evolution of population just based on acoustics. Finally, I want to acknowledge all my partners and collaborators who basically helped me and contributed to the foundation of FADAR, development of all the methods. Professor Henshi Zong at FAU, who's been the chair of the College of the Department of Computer Science, Rick Nemeth at UVI, Rick Appledorn at UPR, the team of the Grouper Moon project, at Scripps, and also the Department of Environment, Cayman Island Government, Alejandro Acosta and Morley, Daniel Morley at FWC in the Florida case, who took us on the Nancy Foster to do some survey around Riley's hump. And then Stuart Fulton at Kobe worked with us, and we work with and basically help us basically spread the word about FADAR and have more people use it. And that's it. And thank you.
Thank you for a great presentation. And now I'm going to take control once again. All right. So if you have any questions, you can type them in the question box, or you can ask the questions aloud by clicking on the hand icon. If it's red, that means your hand is raised, and then you'll need to click on this red microphone, and once it's green, that means you are able to speak. And with that, please raise your hand or type your questions into the question box, and we'll read those aloud. And while people are developing some of their questions, I will ask a question myself. You know, how far do these sound recordings carry from the location of spawning? Do you have to get it right on the spot, or is there some flexibility in the distance that you can be away from it?
Yes, that's a good question. So sound carry based on the source level and the noise in the water. So if it's a very noisy environment, it's not going to carry very far. So we estimated for those species with the sound pressure level, the source level at about 150 dB, you can hear them as far as about 100 meters, and sometimes more, depending also on the substrate, the stratification of the water, the temperature profile, so basically the sound velocity profile. Yeah, sometimes if we've noticed that if you're not exactly at the right location, you may actually not hear all the fish, and that may bias your results because you may record less calls than actually are produced in the water.
All right, I'm not seeing any hands or any questions in the question box, so I'll ask another question. You had indicated that there were 4 grouper species, and then you also had added squirrelfish in there. If any of those individuals are found at a location, do you think that would be a prime location then to look for potential spawning aggregation for multiple species? Or is it better just to focus in on those 4 or 5 species that you already have in FADAR and not go beyond the information that can be analyzed through it?
Yeah, it's mostly because we mostly focus on fish spawning aggregations and spawning site where those species are found. But the reason why we added squirrelfish is that we realized that because it is quite ubiquitous in the recording, it was sort of messing up the classification that we had for the grouper calls. And so we decided to create our own class for squirrelfish in order to improve the performance algorithm. So it's almost like a side effect of trying to analyze grouper sound and realizing that they were kind of messed up by the classifier had the difficulty focusing on just the grouper sound if we did not try to separate the squirrelfish. So I don't think the squirrelfish in itself could be an indication alone that it's an aggregation, spawning aggregation site. It's more like a serendipitous sort of circumstantial decision that we had to make to improve the algorithm.
Thank you. And Texas wrote in, have any of the grouper been frightened away by any of the instruments, or do they not notice them?
So that's the advantage of using acoustics. It's like it's really non-intrusive. So no, they tend to not be bothered about having those hydrophones. They're not really a small form factor, basically, just probably 1.5 feet long and a few inches wide. So really nothing that will disturb them significantly.
Thank you. And then there's another question in the question box. Is there a minimum maximum distance among recorders? For example, if you dispose 3 of them forming a triangle, so you can detect in space where the party is coming, where the noise or party is coming from.
Yes, because you need to hear the same sound on all 3 recorders. So that's really a challenge. So you have to sort of estimate what is your average transmission loss or detection range, your detection threshold on your recorder, and that will determine how far you can put them from one another.
Yeah, it might be influenced by the the sound frequency of the call as well, how quickly it's moving through the water. Is that right?
Yeah, exactly, and how fast the sound would be attenuating or attenuated over distance. So if you only hear the same sound on 2 recorders, then you can really do a 3D or 2D localization, right? You might be able to say how far it is between the two. So you need 3, then you have to make sure that you hear all 3 of them. So sometimes that's why you may want to have more than just 3, but an array of 5 or 6, so that you cover as much ground as possible, and you're able to collect the same sounds on as many hydrophones as possible.
And I said, thank you for the response. So you had mentioned that for some species that are not in FADAR, you might be able to use an unsupervised clustering technique. How many samples would that take in order to develop a cluster to identify that species?
You know, if we look at the number that we use for some of the species, is those numbers are around, you know, 10,000, between 5,000 and 10,000. That's what we use, and to get the accuracy that we have. So then now, depending, you know, on the source levels or how loud they are, depending on the environment, whether it's noisy or not, you may require even more samples to increase the robustness of your algorithm. So that really vary, and the type of call as well, whether it's, you know, it's if it's a loud call or really a subtle call as well. You may have to increase your numbers according to your performance. So that can vary, but what we've used between 5,000 and 10,000 samples. Yeah.
Thank you.
Mm-hmm.
Any other questions? I'm not seeing any hands, so I'll ask another one. We do have a fishery independent survey that's done over here on the South Atlantic coast that utilizes some video cameras, GoPros, and they do capture sound on that. But is the sound captured through a GoPro, would that be sufficient to be able to do some of these algorithms, or is the sound quality in those a little bit not precise enough in order to develop some of these spectrograms?
I think it would worth a try. I'm not sure what the sampling frequency of the GoPros are, but I think it's relatively high. So since those fish produce sound really in the low frequency, they should be properly captured on the GoPro. As long as the frequency, the sampling frequency is greater than 10 kilohertz, then that should be doable. I heard that, yeah, on all the traps that you guys deployed, you have those GoPros, and we can definitely, I think that's doable. Yeah.
Excellent. So another question, any tips, pros, cons on cheap, accessible equipment such as audio mouth you mentioned?
You know, we've been working a lot with the low-grade instruments, which are really, actually, you know, like any other hydrophone market, they're all about in the same price range. I know there is an effort to, and there is, I don't remember the name of the group, but there's definitely some people out there trying to develop much lower, cheaper hydrophones that can be used. So, but you know, I would have to look into it. I don't remember on top of my head. But yeah, there's definitely an effort out there that is trying to do that, to make it available to give greater access to people.
Thank you. All right, well, not seeing any other hands. I'll give it just another 5 seconds. Well, thank you so much for the presentation today. It was extremely informative, and I'm glad you've gotten an accuracy rate. That is amazing to get it up over 90% for these species. So this work is definitely to be commended, and going through these audio files is not easy. So we're very grateful that you develop an algorithm like this, and that can be used to inform fisheries management and hopefully improve management for some of these species. So it's greatly appreciated, and thank you for presenting today.
You're welcome, Chip. Thank you for the opportunity. I'm happy that you guys liked it and see the value of it.
All right. Well, you all have a great afternoon, and thank you for attending the seminar series today.
All right. Thank you, everyone. Bye.